Hello, biology. This is Paulus here. The purpose of this video is to give students an opportunity who couldn't keep up for the 14.1 notes or who were absent during those notes to get an opportunity to finish them up. Happy St. Patrick's Day, even though it's definitely not St. Patrick's Day when you're watching this video. In any event, if you choose to mute me, I won't take it personally because I won't know. But if you do mute me, you will know that you are going to the next slide, or I'm going to the next slide when I point, and there will be a couple seconds delay. Of course, during the video, I will be going a lot faster than I do in class because you can pause the video to give yourself plenty of time. All right, so without further ado, here we go. Getting into human genetics and human traits and human chromosome or abnormalities and human mutations. All right, so remember, CHR stands for chromosome. And what scientists do is during meiosis or mitosis, when the chromosomes are visible, they squish the nucleus, and then the chromosomes are laid out, and they're 46, and they're all mixed up. What they do is they take pictures of those, and then they rearrange them in pairs, and that is what you call a karyotype. And in a karyotype, or the human karyotype, most of the chromosomes look pretty much identical. The only difference is the X and the Y chromosome. The X chromosome is significantly bigger, and it carries numerous genes. The Y chromosome is much, much smaller, and it carries very, very, very few genes. And because the Y chromosome does not carry very many genes, that has a lot to do with why men are expected to have to show sex-linked traits, and we'll get into that either 14.1 or 14.2. I don't remember. All right, moving ahead. Human chromosomes, there are two types. There are autosomes. Those are chromosomes 1 through 22. And then there are the two sex chromosomes, which are the X and the Y chromosome. And we just simply call those sex chromosomes. Sorry about all the noise and the goings on. I just got back from campus to get my classroom ready. And stuff's everywhere. All right. Okay, human gender. So I like to call the sperm that is carrying the X chromosome the girl sperm, and the chromosome that is carrying the Y chromosome the boy sperm, because if it is the X sperm that fertilizes the X egg, it's a girl. If it is the Y sperm that fertilizes the X egg, it's a boy. So that is why you have a 50% chance of having a boy or a girl every time you choose to have children, because there are approximately as many girl sperm, X sperm, as there are boy sperm, Y sperm. Moving ahead. All right, so this is what it looks like in a Punnett square. So, of course, the X and the Y get separated or segregated during gamete formation. And, of course, if we're talking sperm and egg, we're talking gametes, all right? So you got the X and the Y. The female, you technically could cross off this. You technically could get rid of this X because, remember, same allele, same chromosome from one parent. You get to cross it off. And really, it reduces this fraction from 2/4 to 1/2. It's going to be 50% in either case. It would reduce the ratio of 2:2 boy to girl to 1:1 boy to girl. Remember, whenever you are doing a ratio and you have 2:2, 3:3, 4:4, it always can be reduced. And by getting rid of this X, essentially you are reducing the fraction, reducing the ratio at the same time. Moving ahead. Please use this as an opportunity to practice with your notes. Scientists analyze chromosomes by looking at pictures of the complete set of an organism's chromosomes. What do we call that picture of a complete set of chromosomes? Take a look at number 2 if you can't remember. Pardon me. Yeah, you bet. Karyotype. Chromosomes 1 through 22 are called, what's the name for 1 through 22? What are the names of the two sex chromosomes? Yeah, so 1 through 22 are autosomes, and the sex chromosomes are the the X or the Y chromosome. Half of the sperm are male and carry the, while the other half are female and carry the. Take a look at number 4. And I know it's odd to refer to sperm as female, but they will lead to a female if they are involved in fertilization. So the male sperm are the Y sperm, and the female sperm are the X sperm, or carrying the X chromosome. Okay, how do we study human traits? So we use a tool similar to Punnett squares. We use Punnett squares to predict outcomes of offspring. We use pedigree charts to show how traits or characteristics move through families. And you can even predict potential genotypes. That's, remember what GT stands for, genotypes with a pedigree chart. Whoops, feels like I'm slouching. I don't mean to lower my chair either. All right, moving ahead. Okay, so this is a pedigree chart. So squares are males. Circles are females. This line indicates that they have children. Okay. This line indicates all of the children. This female is marrying into the family. This male is marrying into the family. If it's unshaded, it means it is not showing the trait. And again, we use pedigree charts to do simple dominance versus recessive. This indicates a marriage. And if it's shaded, it indicates that it does have the trait. And your goal, whenever you're using pedigree charts, and actually we probably already have done the lab on this, but what you want to do is you want to fill out as many genotypes as possible. This doesn't have the trait. And we're talking about simple dominance. This means this is homozygous recessive. Little a, 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 and little a, little a. Now, dominant traits could be homozygous dominant, or they could be heterozygotes. And you can't know unless you know something about the parents of the individual or about the children of the individual. For example, this one's little a, little a. One of the little a's came from the mom. The other one had to come from the dad. That means dad is heterozygous. Let's see. This one's going to be little a, which means that it had to get a recessive allele, a little a from dad, had to get one from mom, which means both mom and dad are heterozygous. Now, we don't necessarily know whether or not this one's homozygous dominant or heterozygous because there's a 1 in 4 chance that it's going to be homozygous dominant, because remember, this is big A little a times big A little a. 1/4 chance of being big A, 2/4 or 1/2 chance of being big A, little a. We would have to see children from this organism to figure out if they are big A, little a, or big A, big A. Okay? All right. Are human traits associated with single genes? Mostly not. Most of the genes in humans are polygenic. Typically, evolved species have more polygenic genes than less evolved species. For example, humans are much more evolved than a starfish. Okay, we just are. I mean, we can communicate. We walk upright. We've got vision. Starfish, yeah, they do not walk upright. They do not have vision. They can't see. And they're certainly not as intelligent as humans. This flap is driving me nuts. All right, let's get into human blood types. So in the book, you will see the letters I, capital I used for blood types. That kind of confuses students. So what I've chosen to do is just simplify it. We're going to use the A to be the allele for A blood, the B to be the allele for B blood. A and B are codominant, so AB is AB blood, and then O is the recessive. So these are all dominant, but the O is recessive. And if it throws you off because it's a capital O, you might want to just make it a lowercase o or put a line over this O. Whoa, not bad. How many alleles are there in the ABO? It's 3. So this is a multiple allele situation. Remember, whenever you have a trait that has more than 2 alleles, a dominant and a recessive allele, we refer to it as multiple alleles. So it's A, B, and O. A and B are codominant, and the O is the recessive. Moving ahead. Okay, blood type genotypes. So A is going to be homozygous dominant or heterozygous. B, the same. It's going to be homozygous dominant or heterozygous. AB, okay, is the co-dominant form. And O obviously has to be homozygous recessive. And I happen to be O negative. I am a universal donor, which means that any other blood type can use my blood if they need to. That's why they call me literally every 8 weeks and say, could you come in and donate, please? And at the age of 16, you can start donating blood. So I encourage those of you who are giving and who like to volunteer and do good just because it's good to donate blood. Obviously, you're going to have your parents to consent, and you might not do it every 8 weeks, but do it as often as you can. Oh, my bad. Moving ahead. Okay, a blank is a chart used to study human traits. What do we call that thing? You might want to take a look back at number 6. Yeah, it's called a pedigree chart. Most human traits are controlled by blank genes. These traits are called blank traits. If you're not sure, take a look back at number 7. Yeah, 7. Many genes, polygenic traits. If the genotypes for A blood are big A, and big AO, what are the possible genotypes for B blood? For O blood? Take a look at number 10 if you're not sure. Okay, possible genotypes for B are big B, or big B, big O. O blood is going to be homozygous recessive. O. Go Ducks. Okay, let's get into genetic disorders. Many are caused by autosomal recessive alleles. So we're talking about lowercase letters, recessive alleles, okay, found on autosomes, which is chromosomes 1 through 22. Here are a couple of examples: albinism, cystic fibrosis, galactosemia, phenylketonuria, and Tay-Sachs. Moving ahead. Disorders that are caused by a dominant allele, which makes them a little bit more probable, because remember, if you have a heterozygote and a heterozygote, 3/4 of the possible offspring are going to have the dominant allele, which means that they would have the disease. Let's see, this is achondroplasia, Huntington's disease, and hypercholesterolemia. Trust me, I've pronounced these before. I'm just showing off a little bit. These are not the first time I've seen these words, and some of them are a little difficult to pronounce, like achondroplasia. By the way, if you ever happen to meet a dwarf, don't call them a dwarf. Call them a little person. They get very angry when they are referred to as dwarfs or midgets. I do not speak from experience. I just know that for a fact. All right. Moving ahead. A disorder caused by a codominant allele, sickle cell. Sickle cell is pretty common in African Americans, 1 in 500. Disorders and small DNA changes. So remember, small DNA changes in the DNA, change in the letter code or the nucleotide sequence are referred to as mutations. So in cystic fibrosis, which we're going to call CF, and sickle cell, SC, a small change in the DNA of one gene affects the structures of proteins, which leads to the serious disorder. So one gene, couple changes in the DNA, just one gene leads to serious problems. Because remember, genes code for proteins. And if that protein is important or it has an important function, then it can lead to some serious problems. This hat is driving me nuts. I don't want to flip it up like this because then you can't see the little shamrock. Let's find a happy medium. You're like, Paul, let's just get on with it already, will you? Okay, moving ahead. Okay, please use this as an opportunity to practice with your notes. Many genetic disorders are ones caused by recessive blank alleles. Take a look at number 11 if you can't remember. Autosomal, which means they're on chromosomes 1 through 22. Good. A disorder caused by a dominant allele. Give me an example. One caused by a codominant allele. Give me an example. Take a look at number 12 and 13. So dominant would be Huntington's. Codominant would be sickle cell. Uh-oh. Oh, I'm such a perfectionist when it comes to stuff like this. Sorry. Come on. Come on. You guys are like, it really doesn't matter. Yeah, it does. It matters. It matters to me. That's better. How many genes were affected in organisms that have sickle cell disease and cystic fibrosis? Take a look at number 14 if you can't remember. One gene. One gene that codes for a really important protein, and if there's anything wrong with that protein, it leads to a disorder. Moving ahead. Okay, cystic fibrosis is caused by a recessive allele. You guys learned a little bit about this in cracking the genetic code. Remember the baby and also the adult cystic fibrosis, they have mucus that builds up and it clogs the lungs and breathing passages. So it's a 3 DNA base deletion. So the CFTR protein is membranes have protein channels in them that allow things to move in and move out of the cell. And in this case, the CFTR protein is a chloride ion channel in cell membranes. And cell membranes can't allow the chloride ions to move through. Which leads to mucus forming. So because the CFTR protein is mutated, chloride ions cannot move through. They build up, and you get a mucus that forms. Moving ahead. All right, sickle cell. These are bent and twisted shape of the red blood cells, which we will abbreviate as RBCs. How are they changed? So hemoglobin is, by the way, hemoglobin is a very fun word to say. It's near impossible not to smile after you say hemoglobin. Give it a try. Hemoglobin. It just rolls right off the tongue. Hemoglobin. Anyway, hemoglobin is a protein that carries oxygen. One DNA base change, so this is a substitution mutation, causes the hemoglobin to carry less oxygen, and the red blood cell changes shape because of this. So because it's not able to carry as much oxygen, it has something to do with the ratio or the chemical balance in between the environment and the insides of the red blood cells. It's really complicated. I don't really want to try to explain it right now. But because it can carry less oxygen, it shrinks into a twisted sickle, which is, the Grim Reaper, the blade on the end of the long. They look kind of like that. They kind of look like a crescent moon. All right, sickle cell characteristics: low oxygen to tissues and organs, pain, swelling, frequent infections are all characteristics of sickle cell. All right, moving ahead. Okay, give me a characteristic of a person with cystic fibrosis. You can do a characteristic that is seen or felt, or you can do a molecular characteristic. So molecular characteristic is the ion channel, the chloride ion channel. Won't allow chloride ions to move through. That causes mucus to build up in breathing passages and the lungs. Characteristic of sickle cell. Oh, also with cystic fibrosis, it is a 3 base deletion that causes that CFTR protein to be misshapen. Sickle cell, that would be the hemoglobin. It's a substitution mutation, causes hemoglobin to be able to carry less oxygen, which causes the red blood cell to change shape into a sickle shape or kind of a crescent shape. Chromosome that is not a sex chromosome is called, you got it, autosome. Whether a human will be male or female is determined by which? Sex chromosome is in the egg cell. Autosomes are in the egg cell. Sex chromosomes in the sperm cell. Autosomes in the sperm cell. Sex chromosomes in the sperm. Remember, if it's an X sperm that fertilizes the egg, you got a girl. If it's the Y sperm that fertilizes the egg, you got a boy. Why? That's just the way it works. Mendelian inheritance in humans is typically studied by, how do we study Mendelian, which is simple dominance versus recessiveness. So whenever you see Mendelian, think of Mendel, dominance, recessive. Yeah, you use a pedigree chart. Individual with a blood type phenotype O can receive blood from an individual with a phenotype. I don't even remember the names of the answer to this. Oh, okay. They can receive blood only from people who have O blood. You don't need to know that on the test. The ABO blood group is made up of 2 alleles, 3 alleles, identical or dominant alleles. A, B, O. 3. All right, ladies and gentlemen, that is it for the notes. Thanks for watching. We will see you in the Zoom.